Thanks. Hello, everyone. Thank you for having me today. I am Mwada Jallul, and I am from Libya. Today, I will share with you my project on a master degree, which was under the title of the impact of CYP2C9 and VKORC1 polymorphism in patient response to Warfarin and Acenocoumarol. Outlines my talk, introduction, aim and objective, method, results, and finally conclusion. As all we know, warfarin is the most commonly prescribed oral anticoagulant medication that used for treatment of many thromboembolic events. But the most problem associated with the warfarin is narrowtherapeutic index and the big inter-individual variation between the patients and the response to the medication. So most of the physicians find challenges to prescribe the right dose for their patients from the beginning of treatment. So the aim of our study was to evaluate the impact of CYP2C9 and VKORC1 polymorphism and patient response during the maintenance therapy with warfarin or Acenocoumarol. In our method, 100 Libyan patients were enrolled, attending the anticoagulant clinic at TMC, taking either warfarin or Acenocoumarol, and on treatment for at least more than 2 months. A questionnaire was administered and retrospective clinical reviews were performed. Maintenance therapies means the patients on the same dose regimen of the drug at least 1 month based on the INR measurements. I mean, over the past 2 INR measurements. Then we made the genotyping. Fast we made the DNA extraction. Then we screened our selected SNPs. For CYP2C9, we screened CYP2C9 star2 and star3, and for VKORC1, we screened G2A and C2T using high-resolution melting technique or HRM. The last step was DNA sequencing using Sanger method to confirm our results of HRM. In our results, for VKORC1 gene, G2T and G2A, there is a significant difference and there is significant decrease in the mean weekly dose of the drug for the variant alleles in comparison to the wild type, with the p-value 0.02 and 0.04. For CYP2C9, there is not significant difference in the dose between two groups, wild type and heterozygote, with p-value 0.1 and 0.9 respectively. For the impact of non-genetic factors on drug dosage variation, there is not significant difference in the dose between different age groups with p-value 0.14, and there is not significant difference in the dose between different body mass index groups with p-value 0.56. For multiple regression analysis, the variability explained by this model was only 8.6% using genetics and non-genetic factors. That means that another factors may have impact on this big variation between the patients of the response to the oral anticoagulant. In conclusion, our present data showed unlike VKORC1, CYP2C9, star2 and star3 have insignificant effects on drug dosage requirements. My acknowledgements to my supervisors, Dr. Aisha Dujani, Dr. Abdullah Bashin, and Dr. Alhamid Dafi, and to the laboratory of CDC where this study was done. And thank you for your attention.
Thank you so much for your presentation. It was great. Do we have any questions from the audience? I have a question. Are you going to look at any other genes or variants to see if they have an effect on the patient's response?
No, we didn't screen the whole gene, but our further study recommends to for making a whole gene scanning, especially for CYP2, because type 2 star2 and star3 have very low allele frequencies in whole Africa in general. But for now, we didn't make a whole scan for that.
Okay, thank you. And then there's another question. Is there the potential for other CYP alleles to affect dosing?
Excuse me, I didn't understanding the question.
George was asking if there could be any other CYP alleles that could affect the dose.
Yes, some studies shows age may have an effect on drug dosage requirements, but for genes, there is only CYP2C9 and VKORC1 have impact on oral anticoagulant dosage requirements. There's no other genes. But for the another factors, you mean management factors, just age may affect warfarin dosage requirements. Hope I answered that question properly.
Great, thank you. Are there any other questions? George says yes, you answered it. If there are no other questions, then I think it's time for our break.